In today's lecture, we're going to talk about carbon and the molecular diversity of life. During the lecture, minimize distractions by turning off music and listening in a quiet space. Before the lecture even begins, read chapter 3 in your textbook and make notes. Don't highlight, but make notes in your own words in order to maximize your learning. Be sure to take plenty of notes during the lecture, again, putting these in your own words, and then it may serve you best to make an outline before you even listen to this lecture and fill it in as you go. After you're done reading the chapter and listening to the lecture, use your assisted reading questions as a way to figure out what you know and what you need to study a little bit more. You can also use the questions in the chapter in the various subsections, as well as the questions at the end of your textbook. So now that you have all that completed, let's turn off your phones and get started. Here's our roadmap for this lecture. We're going to look at the molecular diversity of life, and we're going to break this down by looking at two subcategories of these molecules. We're going to look at macromolecules and lipids. So let's get started with the lipids. Lipids are not macromolecules. They are a totally other class of molecules that make up the molecular diversity of life. They have a wide range of structure and function, again, that theme that we introduced in that first lecture. And with these wide ranges of structures and functions, they have a lot of different purposes, but their major function is energy storage. Lipids are nonpolar, hydrophobic, and consist primarily of carbon and hydrogen. The first type of lipid that we are going to discuss are saturated fatty acids. The linear structure, which is shown in the figure on the slide, of fatty acids allows the chains to pack closely together. There are no double bonds, so there are more hydrogens in the chemical structure, thus they are saturated with hydrogens or saturated fatty acids. As a result of this structure, saturated fatty acids are solids at room temperature. And an example of fatty acids, solid saturated fatty acids, are mammalian fats such as lard or butter. Unsaturated fatty acids are also fatty acids, but unsaturated fatty acids have one or more double bonds and therefore have fewer hydrogens. So they're unsaturated with hydrogens. You can observe the double bond in the figure shown on the slide and how this double bond results in a kink in the chemical structure. Because of the double bonds, unsaturated fatty acids are often liquid at room temperature and are referred to as oils. Examples are fish oil, cod liver oil, olive oil, and coconut oil. The second lipid we will discuss are the triglycerides. These are lipids found in human blood. The brain sends chemical signals to release fatty acids through hydrolysis reactions to release fatty acids from glycerol when energy is needed. The figure on the slide shows the dehydration reaction resulting in the formation of a triglyceride from glycerol. Remember that all chemical reactions are reversible, so to release the fatty acids, a hydrolysis reaction is necessary. A third type of lipid is the phospholipid. It has a unique structure with two hydrophobic tails and a hydrophilic head. As seen in the figure, the two tails are composed of hydrocarbon chains that do not interact with water. They're hydrophobic. And there is a hydrophilic head that interacts with water. Because of this unique structure, phospholipids are a necessary part of the cell membrane. If you look at the part D of the figure on the slide, you can observe that the hydrophilic heads are arranged to interact with the outside of the cell and the inside of the cell, both environments that contain water. The tails then form a layer in between the two heads. This arrangement allows the cell to be selective about what is allowed in the cell and what is not, keeping the cell healthy. The fourth lipid we will discuss today are steroids. These are lipids that consist of carbon skeleton of 4 fused rings which is shown in the figure on the slide. The chemical group attached to the fused rings determines what kind of steroid it is. In animals, cholesterol is a crucial steroid. Other examples of crucial steroids are steroid hormones such as estrogen and progesterone. So for the lipids, we talked about fatty acids, triglycerides, steroids, and phospholipids. Now we're going to focus on macromolecules. Macromolecules are the essential elements of all living organisms. Organic compounds are compounds that are made with carbon, and macromolecules fit into this category because they have carbon in their structure. Macromolecules are huge and include carbohydrates, proteins, and nucleic acids. Macromolecules are polymers. So what is a polymer? Polymers are monomers that are connected by a covalent bond that is formed due to a loss of water, a dehydration reaction. When a monomer is repeated to make a polymer, the process is known as polymerization. Monomers are connected due to a dehydration reaction, which is shown in Part A of the figure on the slide. All chemical reactions are reversible, so to break down a polymer into its individual monomer sets, the reaction is a hydrolysis reaction, which is shown in figure B on the slide. So if you add a water group, you can break down the polymer. Let's start with our discussion of macromolecules by considering the carbohydrates. Carbohydrates are sugars and polymers of sugars. The simplest carbohydrate are monosaccharides. Carbohydrates are in the food we eat and are the major nutrient of the cell and provide structural support for the cell. In order to connect two sugar monomers to form disaccharides, polysaccharides, and oligosaccharides, there is a glycosidic linkage that occurs between the two monomers. The monomer type and the position of the bond determines the structure and the function of polysaccharide. Monosaccharides are a single sugar monomer. The most common monosaccharide is glucose. Its primary function is to provide energy to the cell. Glucose is converted to ATP during cellular respiration. Monosaccharides are building blocks through glycosidic linkages for other sugars. The figure shown on the slide demonstrates the chemical structure of two monosaccharides, glucose and fructose. Disaccharides are composed of two monomers of sugars. The figure on this slide demonstrates a glycosidic linkage between glucose and fructose, which forms sucrose, the most common disaccharide. Polysaccharides are composed of multiple monomers. They have several functions, all based on structure. Common roles include energy storage, cell structural support, cell-cell recognition, and synthesis of other molecules. One of the major polysaccharides is glycogen. This is a polymer of glucose monomers. Many vertebrates store glucose in glycogen in the liver and muscle cells. Hydrolysis reactions release the glucose when the body requires it. Glycogen stores are depleted within a day unless replenished by consuming glucose. Another major polysaccharide is starch. Starch is a polymer of glucose monomers as well. Plants use starch to store glucose for energy. Finally, we are going to look at cellulose, which is an important structural polysaccharide. It is one of the most abundant organic compounds found on Earth. It is a polymer of several hundreds or thousands of glucose molecules. Its primary function is structural support of plant cells. So we've talked about lipids and macromolecules. We've started our introduction of macromolecules by covering the carbohydrates, introducing monosaccharides, disaccharides, and several types of polysaccharides. Next, let's discuss proteins. Proteins are a biological functional molecule, meaning they serve a purpose in an organism. All proteins are made from 20 amino acids. Proteins are one or more polypeptide chain of amino acids. Proteins are folded and coiled into a 3D shape that determines the protein's function. Humans have over 10,000 proteins, each of which is expressed at different levels at different periods of human development. All the proteins in a given organism at a given developmental time point is called a proteome. So again, proteins are biologically functional molecules, meaning that they have a purpose in an organism. In this figure, there are 8 key functions that proteins regulate within an organism. First, some proteins are enzymes that accelerate necessary biological chemical reactions. Second, there are some proteins that serve as storage proteins, storing necessary energy molecules or building blocks for further chemical reactions. Third, there are hormonal proteins that regulate organismal activities through chemical messages. Fourth, there are proteins that regulate movement. Fifth, there are proteins that are defensive and protect against disease. Sixth, there are transport proteins responsible for the transport or trafficking of substances through a cell or throughout an organism. Seventh, there are receptor proteins that respond to chemical and electrical signals in a cell in order to regulate a response. And finally, eighth, there are structural proteins that regulate the support of the cell as well as transport systems within the cell. Proteins are one or more polypeptide chain of amino acids. Proteins are folded and coiled into a 3D shape, and this 3D shape determines the protein's function. So what are these amino acids? Amino acids are composed of an amine group, a carboxylic acid group, a side chain specific to each amino acid. There are hundreds of amino acids, but there are 20 that are the most common because of their specific role in protein production. There are 4 ways to classify amino acids: the functional group location, which carbon it's on, the pH of the amino acid, the polarity of the amino acid, or the type of side chain that's found on the amino acid. So amino acids are composed of an amine group, a carboxylic acid functional group, and a side chain specific to each amino acid. Shown in the figure here on the slide are the 20 most common amino acids. There are 9 nonpolar side chains or hydrophobic amino acids: glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. There are 6 amino acids with polar side chains, making them hydrophilic. They are serine, threonine, cysteine, tyrosine, asparagine, and glutamine. There are 2 acidic amino acids with a negative charge: aspartic acid and glutamic acid. And there are 3 basic amino acids with a positive charge: lysine, arginine, and histidine. And remember, it is a string of these amino acids bonded together that makes up proteins. Proteins are one or more polypeptide chain of these amino acids. Proteins cannot exist as a long string of amino acids because they couldn't fit into the cell to perform their function. Proteins are folded and coiled into a 3D shape. The primary structure of any protein is the sequence of amino acids that are bonded together. These amino acids can then be coiled together to form an alpha helix or a beta sheet. That is the secondary structure of the protein. Then they fold in on each other in a very specific way to form the tertiary structure of the protein. Finally, the quaternary structure of the protein is the association of 2 or more polypeptides to form the final functional protein. These 4 structural levels of proteins is demonstrated in the figure above. Understanding how the structure of a protein is necessary for its specific function is a key biological concept. Because this is such a key concept, I've repeated the information from the previous slide in a different way. The primary structure of any protein is a chain of amino acids called a polypeptide chain. The secondary structure is formed when the polypeptide chain forms with hydrogen bonds to create either an alpha helix or beta sheet. The tertiary structure is a 3D shape resulting in further folding of the alpha helix or beta sheet. Finally, when multiple polypeptides bond together, the final quaternary protein structure is formed. Here's yet another view of this important concept. On the left is the primary structure, the linear strand of amino acids forming a polypeptide chain. On the right are examples of the secondary structure, an alpha helix and a beta sheet. The secondary structures are formed by hydrogen bonds. The tertiary structure is a 3D shape of the polypeptide chain when the helix and/or sheets fold in on each other. And the quaternary structure is the aggregation of different polypeptide structures. While hydrogen bonds are responsible for the secondary structure, there are several bonds that are responsible for the tertiary structure, including hydrogen bonds, disulfide bridges, ionic bonds, and van der Waals forces. So far we have talked about the steps towards building the secondary and tertiary structures, but there are certain reactions that break down these structures. Breaking down of the secondary or tertiary structures can result in a protein being in its primary structure, polypeptide chain of amino acids. This does not destroy the protein, but just breaks the protein down into this primary structure. This entire process is called the denaturation reaction. You can denature proteins with pH, high temperatures, and certain chemicals. Denaturation is reversible, and a primary structure can undergo a renaturation reaction to form its secondary or tertiary structure. So how can we determine what the secondary, tertiary, and quaternary structures of a protein are? And why should we want to know what these structures are? We need to understand structures of proteins to better understand their function. Function and structure are tightly linked, as you've now heard about in 3 different lectures. One way to determine protein structure is through X-ray crystallography. You can crystallize most proteins and then send an X-ray through that crystal. A digital detector measures the pattern in which the X-ray is diffracted. This indirectly tells us about the structure of the protein. So of the macromolecules, we've covered carbohydrates and proteins. Let's talk about the last molecules, nucleic acids. Nucleic acids, DNA and RNA, regulate gene expression. We need both for proper expression of all our genetics. DNA is what encodes our genes. As seen in the figure, DNA stays and always stays in the nucleus of the cell. So in order for a protein to be made, DNA makes RNA, which can leave the nucleus and contains the genetic code for the protein to be made. The mRNA goes to the ribosome, and that's where the protein is made. It's in the ribosome that the RNA is translated into the polypeptide strand of amino acids. DNA is transcribed to mRNA in the nucleus. mRNA leaves the nucleus, goes to the ribosome, and is translated into protein in the ribosome. DNA transcribed to RNA, translated into protein, is known as the central dogma of biology. This is a key process to understand, and we're going to mention it in several lectures. So what are DNA and RNA made of? Both of these nucleic acids are polymers of nucleotides, which are a nitrogen base, a pentose 5-ring sugar, and a phosphate group. When you're describing the nucleotide without the phosphate group, it's known as a nucleoside. So both RNA and DNA are polymers of nucleotides, the nitrogen base, the pentose sugar, and the phosphate group. Both of these nucleic acids are polymers of these nucleotides. In DNA, the sugar is a deoxyribose sugar. The D in DNA stands for deoxyribose. In RNA, the sugar is a ribose, so the R in RNA stands for ribose. There are 4 bases for DNA and RNA. DNA has cytosine, thymine, adenine, and guanine. RNA has cytosine, uracil, adenine, and guanine. Based on structure, these bases can be divided. Cytosine and thymine and uracil are pyrimidines, and adenine and guanine are purines. As we look at the secondary and tertiary structures of DNA and RNA, these classifications of pyrimidines and purines will be necessary to understand. Biologists determined the secondary structure of DNA using X-ray crystallography, which we just mentioned. Through this method, as well as varying chemical reactions, it was determined that DNA is a double helix and the strands are complementary. The backbone to the DNA is the pentose sugar and the phosphate, so the nitrogen bases are inside the helix. For these complementary strands, only certain bases will line up and bond with other bases. These are known as the base pairing rules. A's or adenines always pair with T's or thymines. And G's always pair with C's. So guanine always pairs with cytosine. So adenine, a purine, only hydrogen bonds to a thymine, which is a pyrimidine. If you know one strand of the DNA, you have an A, then you know on the complementary strand of that DNA, you'll have a T. And the same is true for the C's and G's, the cytosines and the guanines. Cytosine is a pyrimidine, and it will only hydrogen bond to guanine, which is a purine. Because of the double helix structure and the structure of these bases, the purines cannot bind to themselves and the pyrimidines cannot bind to themselves. The biologists trying to figure out the structure of DNA were aware of that. So they had to figure out which pyrimidine bonded to which purine. And in order to do that, they had chemical reactions. And in those chemical reactions, they figured out that there were equal amounts of adenine and thymine and equal amounts of cytosine and guanine in a given sample. So they concluded, and later evidence supported this conclusion, that A and T are base pairs and C and G are base pairs. So we've talked about the double helix, but with all the genes that our cells contain, the double helix isn't small enough to fit in the nucleus of our cells. So the double helix has to wrap around a core of proteins called histones. When the double helix wraps around the core of histones, this is called a nucleosome. The nucleosome then folds up to make fibers of varying loops and widths. Eventually, the fibers are tightly coiled into the chromatid, one arm of a chromosome, and it is these chromosomes that store our DNA in the nucleus of our cells. So how does the DNA, which is stored in the nucleus, make protein? Well, it uses RNA. RNA has other functions as well, but its main function that we will focus on is in the role of gene expression. We've already mentioned the nitrogen bases that are part of the RNA structure. You have cytosine, uracil, adenine, and guanine. And we've also talked about the sugar that makes up the pentose in RNA is ribose. RNA is single-stranded, and it's composed of the nucleotides of these nitrogen bases, pentose sugars, and a phosphate group. How can we tell the difference between DNA and RNA in a given sample? DNA is double-stranded and RNA is single-stranded. There are chemical ways to tell the difference between DNA and RNA, such as X-ray crystallography. Another difference is in the bases. DNA uses thymine while RNA uses uracil. And again, there are chemical experiments that can be done to determine which base is present in a given sample. Finally, DNA is constructed of nucleotides with deoxyribose, whereas RNA has nucleotides made with ribose. And again, there are different chemical approaches that can be used to determine which sugar is present in a given sample. And by way of a reminder, the central dogma. Both of the nucleic acids we covered today, DNA and RNA, as well as the other macromolecule that we covered today, proteins, are part of gene expression. DNA is transcribed to mRNA in the nucleus of a cell. The mRNA leaves the nucleus and goes to the ribosome, and in the ribosome, the mRNA is translated into a strand of polypeptides of amino acids in order to make a protein. DNA is transcribed to RNA and is translated into protein, and this is known as the central dogma of biology. This process has now been repeated 2 or 3 times within this lecture, so be sure that you understand the central dogma. So by way of review, we're going to look at the structure of each of the molecules that we've talked about today and how it relates to their function.
All living organisms are composed of similar chemical compounds called biomolecules. Biomolecules are organic molecules, meaning that they contain carbon-to-carbon or carbon-to-hydrogen bonds. Methane is one of the simplest organic molecules. Notice that the central carbon atom, by sharing electrons, has formed chemical bonds with 4 hydrogen atoms. Carbon is important for life because it's a very versatile atom. In addition to hydrogen, carbon can bind with a number of other atoms, including oxygen, nitrogen, phosphorus, and sulfur. Carbon can also bond to itself, forming chains, branched chains, and ring structures. It is also possible for carbon to form double bonds with itself, or bond to other elements, such as the oxygen shown here. The carbon chain of a biomolecule is called its skeleton, or backbone. This terminology makes sense because, just as your skeleton accounts for your shape, so too does the carbon skeleton of a biomolecule account for its shape. The reactivity of a biomolecule is mostly dependent on the functional group that attaches to its carbon backbone. A functional group is a specific group of bonded atoms that always has the same chemical properties and always reacts in the same way. The functional group shown here is a hydroxyl group, and when it attaches to this carbon skeleton, the compound ethanol is formed. There are many different types of functional groups. In biology, the 5 that you will probably come in contact with the most are the hydroxyl group, the carboxyl group, the amino group, the sulfhydryl group, and the phosphate group. In these diagrams, the R refers to where the functional group attaches to the carbon skeleton. The presence of a functional group changes the chemical reactivity of the biomolecule. Functional groups, therefore, help determine the identity of the biomolecule. For example, when the carboxyl group shown here attaches to this carbon backbone, a fatty acid biomolecule is formed. When an amino group and a carboxyl group join with this carbon backbone, an amino acid biomolecule is formed. All biomolecules can be divided into 4 classes: proteins, carbohydrates, nucleic acids, and lipids. We'll discuss these different classes in depth in the next few tutorials.
Okay, so today's lecture covered carbon and the molecular diversity of life. We looked at macromolecules, including carbohydrates, proteins, and nucleic acids, and we also looked at lipids. If you have any questions about the material in this lecture, feel free to email your professor, to come to office hours, or to seek out your learning assistant, who is also assigned to this course and can assist you with any questions that you have. Now that you've completed the lecture, combine your notes from this lecture with the notes that you took when you read the assigned chapter for this lecture. Once those notes are combined and you've reviewed them, use the assisted reading questions, the questions at the end of the chapter, and the questions at the end of each chapter section as a way to prepare yourself for the quiz and the test on this material.